Welcome to the Explainer. If you're a focused learner looking to understand exactly how cells survive, you are in exactly the right place. Today, we are entirely reshaping how we think about the cell membrane. Look, it's not some boring static wall just holding a cell together. It is a highly dynamic, life-saving gatekeeper. It's a relentless security system constantly making these split-second decisions about what gets in and what gets kicked out.
All right, let's just dive right into this quote from our source text. The plasma membrane can be considered the edge of life. I absolutely love this because it perfectly frames the cell's constant high-stakes border decisions. You see, everything outside the cell is just chemistry. But inside, that's living order. The membrane is that exact threshold. It strictly controls all inbound and outbound traffic, allowing the cell to discriminate in its chemical exchanges. And that selective permeability is quite literally what makes life possible.
So here's our roadmap for today. 1. The edge of life. 2. Passive transport and osmosis. 3. Data analysis on glucose uptake. 4. Active transport and pumps. And 5. Bulk transport, doing the heavy lifting. Let's get right to it. Section 1. The edge of life. Exploring the fluid boundaries of the cell. So the staple ingredient of this entire system is the phospholipid. And to really get it, we need our first crucial definition, amphipathic. Basically, it's a molecule with a split personality. It has a hydrophilic or water-loving region and a hydrophobic or water-fearing region. Think about why this matters. A cell is full of water and it floats in a watery environment. Because of this dual nature molecular structure, phospholipids just naturally organize themselves into a double layer, a bilayer. The water-fearing tails hide on the inside, completely away from the fluid, while the water-loving heads face out. This actually forces the creation of a stable, sheltered boundary right in the middle of a totally watery environment. But it's not some rigid plastic shell. And that brings us to our next major definition, the fluid mosaic model. You've got to visualize the membrane not as a stiff sheet, but as a fluid. It actually has the consistency of salad oil. The mosaic part? That refers to the incredibly diverse protein molecules embedded right in it. Because the lipid bilayer is fluid, these proteins aren't permanently stuck in place. They shift around sideways within the plane of the membrane, kind of like partygoers elbowing their way through a really crowded room. It's this bustling, constantly moving layer of molecules.
Now, here's where it gets crazy. Look closely at how these embedded proteins act as literal security gates, with massive real-world implications. Take the human immunodeficiency virus, or HIV. To infect an immune cell, HIV has to bind to a main receptor called CD4, but it also desperately needs to grab onto a co-receptor called CCR5. Well, a tiny fraction of people have a slight genetic tweak that causes an absence of that CCR5 protein on their cell surfaces. Because that single receptor protein is just missing, the virus is completely locked out, total resistance. That one incredible discovery led straight to life-saving drugs like Maravirac, which actually work by masking that specific CCR5 gate.
Moving right along to Section 2, passive transport and osmosis, effortless movement across the border. Look, we just can't talk about cell survival without talking about water. Osmosis is simply the diffusion of free water across a selectively permeable membrane. Water naturally flows from where it has a higher concentration, meaning fewer dissolved particles, to where it has a lower concentration, where there are way more dissolved particles. And while water can slowly leak through the lipid bilayer, it mostly uses these specialized channel proteins called aquaporins. And guys, the efficiency here is absolutely mind-blowing. A single aquaporin allows water to rush across the membrane at a staggering rate of 3 billion molecules every single second. 3 billion! So the massive takeaway here is that a cell's internal water balance is completely at the mercy of whatever's floating around outside it. We define this as tonicity. It's the ability of a surrounding solution to cause a cell to gain or lose water. And it depends entirely on the concentration of non-penetrating solutes, things like salts and sugars that can't just easily cross the border, outside the cell compared to inside. Notice the stark contrast between these two environments. If you drop a cell into a hypertonic environment, meaning there are way more solutes outside than inside, water rapidly leaves the cell. An animal cell? It'll shrivel up and probably die. This explains exactly why dumping extra salt in a freshwater lake wipes out the local aquatic animals. Conversely, a hypotonic environment has fewer external solutes. Water rushes into the cell. An animal cell, like a red blood cell, will swell up and pop like a water balloon. But if you're a plant cell, this is exactly what you want.
The rigid cell wall physically pushes back against the swelling water, making the plant cells super healthy and firm, or turgid. It's literally why fresh rainwater keeps your houseplants standing up perfectly straight. But what if you don't have a rigid cell wall and you're stuck living in a totally unforgiving environment? Well, organisms have evolved some brilliant mechanical workarounds for osmoregulation. Take the paramecium. It lives in hypotonic pond water, so water is constantly flooding it. To avoid exploding, it actually evolved a contractile vacuole, which is basically a microscopic bilge pump that continuously forces water back out. Marine invertebrates have it way easier. They live in isotonic seawater, where the solute concentrations perfectly match, so they experience zero net water movement. Meanwhile, you have extreme bacteria thriving in boiling hypersaline hot springs. They utilize incredibly unique internal lipid mechanisms just to ensure their water doesn't get completely sucked out. Evolution always finds a way.
All right, Section 3, Data Analysis, Glucose Uptake, Tracking Transport Rates over Time. Let's actually look at some hard data here. Researchers took red blood cells from a 15-day-old guinea pig and a 1-month-old guinea pig, dropped them in a solution of radioactive glucose, and tracked exactly how much entered the cells over 60 minutes. Now, because glucose is pretty large and polar, it has to use carrier proteins to cross the membrane in a process called facilitated diffusion. And this graph brilliantly illustrates a clear biological trend. Age significantly affects the rate of facilitated diffusion. If you look closely at the 60-minute mark, the 15-day-old cells absorbed roughly 85 millimolar of glucose. In stark contrast, the older 1-month-old cells only took up about 30. The younger cells are just pulling it in so much faster. I mean, seeing data like this forces us to genuinely ask questions about how cell membrane protein expressions fundamentally change and adapt as an organism develops. Which brings us to Section 4, Active Transport and Pumps, Spending Energy to Fight the Tide. We really need to draw a hard line between these two concepts. Passive transport requires absolutely no energy. Molecules just naturally drift down their concentration gradient, totally effortless, like a raft smoothly floating down a river. But active transport is the exact opposite. You've got to pay to play. It requires a real investment of cellular energy, usually ATP. Carrier proteins in the membrane must physically pump molecules against their concentration gradient. We are talking high-energy, uphill heavy lifting here. The absolute classic example of this is the sodium-potassium pump. Let's walk through this precise mechanical sequence. Your cells desperately want a lot of potassium inside and a lot of sodium outside, which goes totally against the natural flow. So, step 1, cytoplasmic sodium binds right to the pump protein. Step 2, ATP slops a phosphate group onto the protein, which radically, physically changes its shape. Step 3, that sudden shape change violently expels the sodium outside the cell, which conveniently opens up new binding sites where external potassium attaches. Finally, step 4, the phosphate group drops off, the protein snaps right back to its original shape, and the potassium is yanked inside the cell. Guys, this pump is literally an electrogenic battery. By pumping out 3 positive sodiums for every 2 positive potassiums it brings in, it generates the actual cellular voltage that powers our nerve cells. How cool is that? And this active pumping sets up massive gradients that the cell can exploit for other tasks, a process we call cotransport. And this isn't just textbook stuff. Let's grasp the very real, very serious impact of this. In developing countries, severe diarrhea causes a life-threatening loss of sodium and water. You can't just give a patient pure water. But by pairing salt and glucose together in a really simple drink, you trigger a specific set of cotransporter proteins in the intestine. The sodium rushes back into the cell down that gradient the cell built earlier, dragging the glucose in right along with it, and water rapidly follows through osmosis. Literally manipulating this microscopic cellular machinery through oral rehydration therapy has drastically lowered infant mortality all over the globe. Okay, on to our final section, Section 5, Bulk Transport. Heavy lifting, moving massive cargo across the border. Sometimes things are just too big for a tiny protein channel. The cell actually has to package these huge items in vesicles. Let's quickly break down the 3 distinct methods a cell uses to swallow bulk material from its environment, a process called endocytosis. First is phagocytosis, or cellular eating. The cell literally reaches out with pseudopodia, like little cellular arms, around a large particle and completely engulfs it. Next is penocytosis, or cellular drinking. Here, the membrane just continuously pinches inward, gulping tiny droplets of extracellular fluid and whatever random solutes happen to be dissolved in it. Finally, we have receptor-mediated endocytosis. This is the VIP line.
The membrane uses targeted surface proteins to grab hold of specific, rare molecules before pinching inward, allowing the cell to acquire bulk qualities of exactly what it needs. Let's anchor this in a serious medical reality. Human cells use that receptor-mediated endocytosis to pull cholesterol right out of the blood. The cholesterol travels in LDL particles, which bind to specific LDL receptors on the cell membrane, basically telling the cell to swallow them up. But in an inherited disease called familial hypercholesterolemia, these LDL receptors are either defective or just completely missing. The cholesterol literally cannot enter the cell border. Consequently, it just dangerously accumulates in the bloodstream instead, leading to early severe atherosclerosis, clogged arteries, and potential heart damage. A microscopic breakdown with massive consequences.
Now, the exact reverse of this process, shipping large cargo out, is called exocytosis. Let's trace the biological path. It starts deep inside the cell when a transport vesicle carrying a huge payload buds off from the Golgi apparatus. Second, this vesicle actually travels along a literal highway of cytoskeletal microtubules straight toward the edge of the cell. Third, the vesicle contacts the plasma membrane, and because they're both fluid-lipid bilayers, they just smoothly fuse together. Finally, the contents spill entirely out into the extracellular space, while the vesicle's membrane permanently becomes part of the plasma membrane. This exact mechanism is how organs like your pancreas successfully export massive, life-saving waves of insulin into your bloodstream. So we've covered a massive amount of ground today in this explainer, all the way from the fluid mosaic model to complex data tracking glucose uptake. But I want to conclude with this one provocative thought. As you sit here watching this, inside your body right now, billions upon billions of cellular gates are violently pumping ions, gulping fluids, and actively keeping your chemical balance perfectly in check. The border is alive. What would happen to you in the next 60 seconds if your fluid mosaic suddenly just locked shut? Well, you wouldn't last a minute. That relentless, microscopic gatekeeping machinery of the membrane is literally keeping you alive this very second. Thank you so much for learning with me today, and keep exploring.